Good afternoon to everybody. We are welcoming Dr. Christina Kuchendorf from Germany, and it's our pleasure to invite you as a keynote speaker in our conference organized by the Palestinian Society for Biological Sciences with coordination with Palestinian Academy for Science and Technology. Let me introduce Dr. Christina is a researcher and projects coordinator at the Institute of Bio and Geosciences, IPG2, at Forchungszentrum Juelich in Germany. Having graduated with geobotany and computer science and started out in a joint field projects in China and Juelich, and later in Montpellier, France, with the main topics of plant nutrition and resilience against flooding and other stress. Since 2030, back in Juelich, algae were her focus, first with assessment of the added value chains of microalgae lipids productions to jet fuels, etc. And since 2016, with a coordination with the IPG2 Algae Science Center with 3 pilot-scale algae protection facilities, control and study of microalgae productivity and growth dynamics in large volume cultures have since then been a major topics. The ultimate target is to simplify algae carbon dioxide sequestering for broader accessibility and better connectivity into industry, agriculture, and city environments towards autonomous intelligence algae culture control, and the technical implementation of modern bioeconomy. Last but not least, focus is the usage of algae in plant nutrition, especially the possible benefits of algae, algal biomass, and other organic fertilizers in improvement of poor soils and nutrient cycle, always aiming to keep science applicable and working towards circular bioeconomy. Her talk will be enabling an intelligent, sustainable, and future-oriented bioeconomy from problematic wastewater to circular water management with the help of algae. So, Dr. Christina, it's our pleasure to hear you, and it's your turn now. You are welcome.
Thank you very much. I'm sorry, I did not know how short or long the information had to be. Can you tell me if you can see my screen? Can you see the presentation?
Yes, okay.
Okay, then I'm going to start. So my aim today is, give me a second. So my aim today is actually to provide an overview about some of the uses of algae biomass in economic and possibly bioeconomic processes, and also give some motivation about connectable bioeconomic solutions and why or how to push their implementation in the future, and why this is not only important for people in Germany, but it is really a worldwide challenge where everybody can really profit from everybody else's experiences. So what is bioeconomy seen as actually? So it's a production and the use of biological resources to produce products, also for processes and services in a biological way. It goes towards a resource and climate-friendly lifestyle and work, and towards closed substance and cycle waste management. For Germany, that means that there was a decision of the German government to withdraw from coal-fired power generation until 2038. And also the general fight against the ramifications of climate change. And therefore, large parts of the Federal Republic of Germany are now confronted with the need for structural changes that affects many areas of daily life and pose some major challenges for the people that live there. For example, the people that lived around the great lignite plants. But this also offers an enormous opportunity to bring about a rethink in our fossil carbon-based economy towards another economy which is based on bioeconomic principles. An additional motivation is, of course, the climate change effects. So people here right now are maybe thinking, yeah, why is Europe worrying about water? But this is still a joint motivation to better deal with this resource. Because even in Germany, we are feeling that in the last 3 years, we had 24 months which were far too dry. We only had 11 months which were too wet and which were not at the right time for crops. So we have had quite an experience about what it means to not have enough water for our normal crops. And if the world average temperature continues rising, our areas even might experience like 10-year droughts. Yeah, we are not used to this, so it is a bit scary. So why there is something like further biological wastewater treatment, which you have seen on the starter slide, may be important. There is the question of the additional question of nutrient reuse, especially of phosphorus, during this water providing question. Water is already an endangered commodity in many regions of Europe, as you've seen here, and will even be more under stress in the future, also worldwide. And there is a motivation here that we share also with our current Palestinian partners here. And it's not just a commodity, it is really valuable. And the nutrients which are maybe in there are also valuable. They can be, or there were, and maybe can be again, fertilizers to ensure crop growth, which goes especially for phosphorus and for nitrogen. And while, for example, nitrogen compounds are not scarce, but rather subject to accumulation at the wrong place and the wrong time, for example, in the water, causing eutrophication and posing a danger to water ecology and health. On the other hand, then the phosphorus is a different matter, and for both, a circular use has many benefits. As for phosphorus, phosphorus distribution worldwide is relatively inhomogeneous, and the loss of both phosphorus and nutrient to the water causes eutrophication in water bodies, and the existing wastewater streams contain lots of phosphorus and nitrogen residue. So being able to close these nutrient cycles also has a matter of being locally more independent of global movements of nutrients or water. So this is, of course, also something which is interesting. The natural cycle of phosphorus, for example, takes about 400,000 years, which is an additional motivation to have this impending boom of maybe at some point not enough fertilizer gone from over our heads. So the intelligent and sustainable water and resource management and handling is urgently needed. This does not just go for agriculture, but also, for example, for cities or urban areas with their water flows and nutrient cycles. These are also sources of treated water and nutrients and can even be seen as production units in this regard. And at present, used and nutrient-containing water is either just discarded or maybe disposed to wastewater treatment. And techniques to regain the nutrients here are needed or need to be subject to improvement because they are by far not perfect yet. So these could be enhanced by additional treatments like biological bacteria or algae nutrient uptake. We have even heard about it already today. And this does not necessarily even have to happen in a wastewater treatment plant, but there can be even more local solutions, especially directly connected to agricultural processes. So the target is in the future at some point a true nutrient cycle, especially for phosphorus, to have more independent resources. So here's a picture of this nutrient cycle. I'm not sure if you can see my cursor, I hope so. You can see there is on the left side, there's a rather large arrow of phosphorus which is mined going in. And there are still losses of phosphorus during the use in agriculture, in food and feed and production, where phosphorus ends up in fixed forms in the soil and is not useful anymore. or where phosphorus is lost into landfills and rivers, where it can even be harmful in non-wanted algae blooms for eutrophication processes. At the same time, I just said the word algae blooms, they thrive on these kind of nutrient loss. Algae could, for example, be used to get this stuff back into the cycle, and I will elaborate a little bit further on this on the next slides. So algae as a green resource. We have already heard a lot of bacteria there, but algae are also kind of quite interesting species in this regard. Our actual mineral oil reserves consist largely of very old algae, whose biomass has conserved sunlight energy throughout the Earth history. And under optimum circumstances, they would be able to grow 10 to 40 times faster than higher plants. Therefore, that's a laboratory value. But they are still growing quite quickly also under normal circumstances. Several pretty names, and so far there are more than estimated 200,000 species out there, but only about 100 are right now exploited worldwide. And almost none of them are somehow genetically modified. So people are still working with wild types, which also underlines the potential that these species in theory can have. And of course, they are able to use sunlight for phototrophic growth and nutrient uptake, and they can so therefore also be carbon sequestration thrown into the mix of uses which this biomass has. So there are still some questions needed for applications where you want to use algae, like will they be able to live where they are supposed to work? How weather dependent is the process? Since sunlight and CO2 should be used as an energy source to make it even more sustainable. This is actually a more critical question for Germany, because anybody who has already been to Germany knows that weather is kind of our most chaotic parameter which we have. How productive could the algae then be in any given environment? You need some measure to give this. And these are, for example, questions that are being solved at IBG2 at the current time. Another question is, of course, additional value from this biomass, because they can produce different kind of lipids, contain different forms of sugars, lignite, etc., which are higher, or pigments, which can be higher value products, which could form additional value chain processes. But this is today not part of this presentation. If you are interested in this, I would ask you to contact me afterwards about this. So we have, I have some pictures, so yet you can imagine how algae are worked with and presented today. For example, in fermenters, which is not interesting so for us because we want to do phototrophic things and it is too dark in there and they have to be fed with sugar, which causes an additional use of agricultural land. We do not want this, but it is done and it is efficient if you want to produce certain products. Then there are these so-called open pond productions, for example, on Hawaii or even in Israel, where they are using saltwater and sometimes freshwater cultures. This is the cheapest and most widely used way to produce algae in a certain climate zone. So in our areas, we need more, in Germany, we need more controlled systems, for example, in tubes or in bags, which can look like this, and sometimes even in greenhouses. So currently use of microalgae, just as a general overview, because I will concentrate on the agricultural aspects later. Algae can be used as food additives or colorants, even go into pharma and nutritional products, from shampoo to spirulina as a health booster for you. They can be used for fish feeds. They can be used in theory for biomass and biofuels. For this, the value chains are established, but as long as oil is too cheap, this is far too expensive, for example. And they can also be used in wastewater treatment and nutrient cycling. This is where we are going. Just a side note, if algae are produced in freshwater, there's a very simple connection to have them later used in wastewater treatment because there are no disposal conflicts. We do not want more salt than needed on any agricultural used field or any other substrate for plants. But salt water normally makes it more easy to cultivate them. It's a bit less maintenance. And we have seen in the past, it much also depends on having the right species, which very often are just local species of algae which live in your surrounding anywhere. They know the pathogens. We do not even have to be too specific. And then there are some products like some sweets which use it as colorants, some food additives, pharmaceuticals, some animal fodder, and also here for fertilizing and nutrient recycling and nutrient uptake from wastewater, as you can see in the corner. So some information about how to actually grow them. So we have gained some experiences in algae production on larger scale over the years at Juelich. You can see some type of photobioreactors here on this slide, some outdoor photobioreactors on the lower left, which can be even heated or cooled. Of course, takes some energy. We have a greenhouse where we can produce algae, and if you have them in the greenhouse, you would, for example, centrifuge them to get a nice creamy mass of algae. Then you go and put them into a spray dryer or a solar dryer. And when you want to put them into a soil, you can do this in a powdered formulation or even in a wet formulation, depending on what your target is there. So what can one produce in a location like ours per year? We have finally met, after some years of running in, an average yearly production of about 73 tons per hectare and year. Just for comparison, Hawaii algae plant would, after my knowledge, produce around 30 tons per hectare and year. But it's an this was the open pond, which are generally a bit less productive, but it's a run-in production, which is also very reliable. So what can this biomass then in our wishes be used for? We have done some preliminary experiments a few years prior where we have compared mineral fertilizer, just a normal NPK fertilizer. We have used algae biomass, and as comparison, there is a non-fertilized wheat plant here to see if nutrients can actively be transferred from the algae through the soil to the plant. And the answer was yes, it works. And an additional bonus is, of course, that algae contain not just nutrients, but also carbon and other maybe beneficial components to enhance and boost the soil microbiota, which is also not researched very well yet in this regard. So this was a very interesting way to take this further. Click. Here's something which part of which can actually also be seen in the Natural Museum at Bethlehem. It's the so-called algae turf scrubber. And this is where algae are used for production of cleaner water, where you actually rinse the water over a pathway, which can be, this is an example, it can be much longer than this one and have better effect even. And the algae take up the nutrients from the water, and the product is first clean water. And when they have grown enough, you can harvest the algae biomass, which gives this nice green sludge. And this could then, for example, further be used as a fertilizer. You can ask, for example, Professor Sharaf or Mazen Bousi, who's also speaking here later about this, because there is an installation around. And this kind of system, for example, can put into use almost everywhere where there are nutrient-rich waters. So this is a relatively flexible process. So this is very applicable for us. Another challenge is that we are able to produce all around the year, because of course algae have a favorable light and also a favorable temperature regime. Here, for example, you can see the changes in light throughout the years, which is the yellow line, and all the changes in temperature throughout the years. And these are already average temperatures in this case. And the algae would actually prefer it almost always a little bit brighter and a little bit warmer than Germany has to offer. So this is a kind of, how do you say, if you are getting interested, you would have the place to go. So the algae biomass, which can be obtained there, can incidentally very easily be applied to fields instead of mineral fertilizers. And we believe that a very good contribution to nutrient cycling is possible here. It is not only about the fertilizer and nutrient transfer, though. It is also about the carbon transfer, the improvement of the soil, and about using the best suited resilient plant species. There will be actually more talks following in this session afterwards. It is also about other alternative fertilizing techniques which bring back carbon and nutrients to the soil, and which may be able to improve depleted or desertificated substrates to make them usable for agriculture. All this still in a resource optimizing way towards the circular economy, or here even bioeconomy. So these pictures, for example, are from when we tried out some formulations of algae. Here's something with wet algae. And if you do not really take into account the property of your soil, for example, you can end up in a very fine sandy soil, which is very dry in the beginning, that it will not take up very well this fertilizing solution. If the soil would have been more wet, it would have been easier. So there are many things to learn, and there's also many things to learn from people who are much closer than to agriculture than we as scientists actually are. So there is also a kind of feedback and the kind of networking needed in the way to bioeconomy, to a circular bioeconomy, which cannot be done alone by a single group in this. So what we are doing right now in the framework, for example, of the Palestinian German Science Bridge, and also in the base of a project we have had in the Palgir framework, is we are now targeting marginal fields experiments, where right now 2 PhD students have started in Juelich to work on the question of nutrient transfer and soil improvement by different organic fertilizers. This includes the algae, but this also includes, for example, the combination with biochars and residues from biogas facilities. We are very interested in the dynamics and the transfer of nutrients, which turns out here, and also about the development of the soil in this regard. We have a chance to use relatively empty soils, which are very young and have not been used agriculturally before in the history after the lignite mining, which has been done in our area. So we are very looking forward to dig deeper into these questions. So, so the game is here targeted against soil amelioration and clearing up the mechanisms behind it, and also finding the best way of application. Because in what we have been doing already, we have found, for example, that the timing of fertilization can be very important, and of course also the plant species, because not all of them have maybe the right technique to taking up nutrients. What happens in the rhizosphere, which is the zone where the plant connects with the soil, can be very different depending on the plant species. So each plant has a different plan how to take up nutrients best, which can be changed up to an extent, but not in every direction, and not in a wide range for every plant. So every soil, every treatment, every plant can have the need of a tailored process where there is much to be learned. In traditional agriculture, very much is already learned from experience, from the crops which have been used for a long time. And we are learning from that experience, but with this new soil boosters, which I like to call them, we will have to compare and to try to also learn the new things there. So, but can things like this be the one solution? No, but it can make lots of motivation in many places for many part solutions. Now I'm getting a bit more general again, because what you here see is a picture of 3 cycles in some urban environment and the resources which can be found there. They can also be found in smaller scale in a house or in a farm. I just chose this as an example because everything is on here. So there is some motivation, which is also for the young people to get into motion and realizing these things. One could view 3 resource areas which do not yet overlap very much. So right now there is a part with energy and heat, where there are some resource-oriented processes, like you can use photovoltaics, solar thermals, pellet heating. All of this is rather investment high, so there's relatively high cost. If you go to industry, some things can be connected, like reuse of heat for urban settlements, for example. Then you can go on the blue side to the waste and process water, where you can have, for example, a connection to biogas, or you can separate wastewater from a toilet into the yellow water part and the black water part. That is how it is called. For example, urine, the ammoniac-rich part, and the the biomass rich part, and then use this further to avoid too much load for the wastewater. You can even use rainwater usage, because depending on the area, rainwater can be, when in an area where there's much traffic, relatively nitrogen rich, and can also be used then in the runoff, and you could also retrieve nutrients from there. A relatively small part is the biomass circle, which actually should also be green, but for some reason it's brown. Maybe it has already fermented, I'm not sure. And for example, for biomass, there are larger solutions like sewage treatment or sewage sludge treatment or biogas plants. There are not so many local solutions yet, but in theory, it would be possible to have, for example, for a city quarter, a very small biogas plant, or for a farm to have a very local, very small biogas usage, which can produce digestate and produce some gas to heat a stove or something. So there are some innovative ideas, but there are not so many of those realized yet. And there is not the one big solution, like finding the one biomass which can solve all our energy and food problems, but the future solution will be complex and will be like a puzzle put together from all directions and be very much networking and connecting different resources. In addition, all these, we have now a lot of progress made in machine learning and in connecting different processes. And this will also help a lot to more intelligently combine these kind of processes to get them the most effective. The thing is that all starts on a small scale. There are lots of students actually having very good ideas and trying to set them into motion within these kind of frameworks and the resources which they are possible. But to find out, so there need to be initiatives on many scales to learn about the right magnitudes and to reach efficiency and to overthink the infrastructure, which is where you actually are. Right now, when very many places, it accepts too many losses to enable a true circular economy. So we need to catch those losses. I have an example on the next slide. For example, from 100 square meter roof area, if you were in Germany, which could deliver electric energy during the year of 8,000 kilowatt hours. It could deliver solar heat for about 30% of the total need of heating, which you would need in Germany. Can deliver about 160 cubic meter of water per year. But it could also be an area to produce algae biomass of about 30 tons per hectare per year with 500 liter per square meter water for irrigation or gray water use, for example, for washing dishes or washing clothes. So this could be a balance where one could start up. If I have this balance, okay, where could I connect? Where can I connect to? Which new systems? Where I could find an intelligent solution? What is not yet used? What is lost? Because right now, what is lost most is heat, drinking water, and biomass. Alboraey.
Try to finish, because
especially biomass and nutrients are lost very easily. Also for this, I recommend for very good ideas to visit the Palestinian Institute for Biodiversity and Sustainability, because I've had the chance and it was really good. And for example, some ideas which students have had in Germany in the last year, there was a kind of competition where they tried to get greener ideas. For example, one solution was here, the Loopside project is supposed to connect water users intelligently in an Internet of Things over a whole city to see where things are lost and where they could be used otherwise, just to get an information base. And there is this urban pergola, which just is supposed to bring more green and more shade and easier climatizing. And then there are waste to resource unit, which, for example, can use biological waste or coffee residue and get back substances of value for it. And there was some nice ideas for this. So I hope I can motivate now some of the listeners to pursue, maybe not exactly those, but similar solutions in these fields in the future. Because if more people do it, it gets easier, it gets more affordable, and the knowledge share is improving. We have social medias, we can talk much more easy than years ago. So let's network, let's learn from each other and build a more sustainable bioeconomy everywhere possible. So with this, I would like to thank you for your attention. And if there are questions, by a way of contact me later, I will be happy to.
Okay, thank you very much for your interesting talk. We can, if there is a quick questions from, okay, Dr. Hanan. Dr.
Okay.
Hanan.
Yeah, do you hear me?
Yes, yes, go ahead.
Yeah. Okay, thank you so much, Christina. I mean, this is really, I really enjoyed your presentation, and it's very important topic indeed. I have a question for you. You know, with the CO2 emission and the climate change, as you know, it's really one of the most, or maybe it's the most important crisis nowadays, and not just currently, it's in the future. So how microalgae, you know, and with their capturing of CO2 and fixation of CO2, do you think this could help for the climate change problem, and how even climate change affect these microalgae? Thank you, Christina.
Thank you for the question. I think the crucial word in here is help. Because microalgae will not be the solution for CO2 neutral products. The photosynthesis is not a a very efficient process in that regard, which is, okay, it happens everywhere on the world. This is our only natural fixation process right now. But if you have to employ algae, you also have to use some pumps normally. You have to give them CO2 by bubbling. You will have some loss through the surfaces. And if you produce a kilogram of algae, you will use for that between 2 and 4 kilograms of CO2. So yes, they fixate CO2. The thing is that the processes in which they do that also produce CO2, and they are not good enough yet. Especially when you have to use harvesting techniques like centrifugation. So for sure, these are processes which can help towards a better CO2 balance. And there are processes envisionable where this is actually a positive balance. We have, for example, now some experiments done with a so-called CO2 concentrator, which is a machine which concentrates CO2 from the air and then pumps it through the algae. Which also works and can, for example, be solar powered. But it is not a generally usable technique yet. So, of course, this is biomass which is produced by using up CO2, but the surrounding work is, depending on the products that you want, it needs more development. So what is working relatively well are these so-called turf scrubbers, because they do not need additional CO2, so there's no additional loss also not in the air by bubbling, but they take up the air CO2 in where they are, and they are easy to harvest. So this is, for example, an easier step in the right
Dr. Christina, thank you very much, and we look forward to have much more cooperation between us. And so we will have a general discussion at the end of the session. So please be around, everybody, at the end of the sessions. Right. We can move now for the second speakers in this plant biology sessions. Ahmed Alboraey. Ahmed Alboraey holds a master's degree in agriculture biotechnology from Osman Ghazi University in 2021, and a bachelor degree in biotechnology from the Islamic University of Gaza to 2015. He worked at the Ministry of Agriculture and as a teacher assistant at Islamic University of Gaza. He also engaged in the water and agriculture related issues and take the position of a Blue Peace facilitator in the World Youth for Parliament for Water and theme coordinator at Mediterranean Youth for Water Network. Ahmed Alboraey, he is going to represent his presentation about the Agri-nanotechnology in Palestine challenges and perspectives. So Ahmed, are you here? You can start now.
Okay, thank you, Dr. Sharif, for the introduction, and I also would like to thank you, my colleagues, Mahmoud Hamou and Ghassan Matar, for this work from different background. We have established this one with nanobiotechnology and agriculture biotechnology, and also from water treatment. These are our background that we have came from, and we have engaged in this work. And I would like to share this on behalf of my colleague. So as we know that the agriculture knows that the background, the backbone for all the support system, and merely in the developing countries. But we can find that Palestine has a special issue since they are occupied and they are located in the Mediterranean Sea, but they have a limited water and land resource that go due to the ongoing Israel occupation policy and the climate change that have been affected on the agriculture sector and lead to the decline on the production and the yield. We can find that that during the last report and the strategy that the Ministry of Agriculture has held in 2017-2022, they are starting to work on more initiative and sustainable solution, and they are working to improve the effect, they are working to decline the effect of this change on the land. So we found that nanotechnology could be a new technology that can help the plant health. So with this technology, we can increase the yield and minimize the nutrition loss in the fertilizer, and also we can control the pest and the nutrient management in the future. So with the objective of nanotechnology in the agriculture, we can increase the yield and also reduce the waste and nutrition loss nearly to zero, so we can increase the efficiency on the agriculture sector. But still, this technology needs new development and also still under development and need more technology to be implemented, so we can use it for large and improve the economic for it. But so it is still possible. Improving these technology tools is very important and will be critical in the future. So in this graph, we can see that the nanotechnology in agriculture can be sorted in different level, and we can find the category for these. Most of these would be related for abiotic and biotic stress that can affect the plant, and the other will be, we can sort them like this. Some of these will be related for the soil, which we can also enhance the soil enhancement by nanomaterials, and the nanofertilizer can also improve the crop growth by increase the absorption of the nutrient. And also in the molecular level, we can improve these by have a good and improved species from plant that can be tolerant for the stress that can affect them, and for the control and the biocontrol for the pesticide. The nano pesticides can be used to protect the crop. And for the technology in general, we can find that the nanosensors can give us all the information. We can find and monitor the plant during the situation, if there are any increase on the pH or temperature, or even for the need for water. And in the salt stress also, which can be a stress tolerance, which can be from different reasons, like food, or for example, the increase of the UV that can affect the plant and increase the gross production in the plant, which can be a harmful effect on the plant. So this is the main idea. Methods can be nanotechnology used in agriculture. In the last, in ISSW science, in the last years, we can find that the number of nanotechnology agriculture articles have been published, which are around like 700 articles per year, which represent about 0.5% 5% of all the articles have been published in the agriculture in general. So this is still not high rate for the production, but there will be increase in the future. We think that, because also in the agriculture we can find that there's many other sectors have been working related for policy and water management and other that can be worked to improve the agriculture sector and to reduce the waste of food and reduce the waste and improve the food and zero hunger. The fertilizer and nano pesticide, the Ministry of Agriculture have been starting new rules for registration for fertilizer, and they aim to clarify the use of these fertilizers since they're known to have toxicity and high toxicity. Some of them have high toxicity. So in the last report, they are giving a license for chemical pesticide for the shop. About 150 have been given, and they accept around 300 from entering of pesticides from the other side, 115 around from the abroad, and they register about 35 new commercial pesticides. We can find that this number is low because for the new pesticides, because we find that we have more than what we need from the commercial pesticide that needed for the use, especially in the current time and the decrease of the arable land. One of the catastrophes that happened is that during the last aggression on Gaza Strip, there is a store for pesticide have been destroyed in Beit Lahia that contained 1,000 of pesticide. Maybe it will be, it will have an impact on the life, not on and the environment and the life of the neighborhood in this area. So we thought that we should have a quick response, and we thought that we might need to find a co-friendly product to at least to save our environment and save our humans' lives in this area. So we thought that nano-fertilizer could have the potential to give a neutral ability and access for growing seed, and that can be improve the root and the shoot length, and also could give also a quality and yield of vegetative and improve on the growth and in the chlorophyll content. On the last recently application also find that agricultural field typically consists of encyclopation of well-known herbicide, fungicide, insecticide. These are synthetics, could be from clay, silica, and lignin. These are natural polymers, can be degraded without any effect on environment, and they can be eco-friendly to be used in our country. On the other side, we found that the plant response to abiotic stress under nanoparticle have been improved during using these silicon filterizer had been used to promote the effect of physiological and morphological trait on vegetative feature of basil under salinity stress. There was some improvement in this plant and the tolerance for this stress. Water pollution and environmental hazards on agriculture. As we know that most of these pesticides can lead to the underground water, and most of this water, or we can see most of the water used in the Gaza Strip and West Bank is based on the groundwater, which represent 90% of the water supply. They are polluted and they are associated with agriculture to achieve food security and fertilization. Also, nanotechnology can find the solution for wastewater treatment, and can be these sustainable and low cost. One of these solutions can be like the photocatalyst, nano absorbent, and nano membranes. These could be used like one of these is titanium oxide, could be used and have effective and efficiently way for wastewater treatment. On the other side, nano sensor for precious farming and smart agriculture. This could be used more ICT technology on more something to monitoring and follow up with the plant's need, just like to if there will be any change in the critical parameters such as temperature, pH, and humidity could be monitored, and we can respond quickly for this change, as we can see this plant to have a high yield of the fruit. Many types of these, like they include the fluorescence resonance energy transfer based on nanosensor, carbon-based electrochemical nanosensor, pneumatic nanoware, and antibody nanosensors. These will be just like produced from the plant, as if there any change in the plant cycle or in their signal that can produce due to the stress or any reaction that could happen from any predator. And also we can monitor the soil nutrient and the fertilizer, humidity and pH, pesticide, herbicide, and microbes causing disease, hormones, and overall crop growth rates. This could be also monitored by the nanosensor monitor. So stress tolerance for improvement of the crop production. Agriculture can also improve the crop yield, as several research have demonstrated. Salinity stress, for example, can limit crop production up to 20% of the more cultivated land. To solve this, they have used some types of spray containing for sulfate nanoparticles. These have been applied on sunflower, and they have been found to increase the shoot dry weight and expand the leaf of the plant. This could be used for, as we have some area to increase their salinity and are not able, we are not able to use them for cultivation and agriculture. We found that the conclusion after we study this situation based on our country, Palestine, and the challenge that we found, and what is the possibility for using nanotechnology in the future of in the near future. These are more precisely in the agriculture sector under development and international level, and they will be cheap, and equipment of nanotechnology will be enhanced plant growth and tolerance, but and also there could be a promising solution for the current situation in Palestine, and we definitely will help to merge more technology in the agriculture sector. And we still need more research to be in this sector, and also we should have to work to build the capacity for the farmers and to know how to use this technology so we can increase the yield of the production. And research on nanotechnology. Yes, the last slide, please. On advanced fabrication, characterization, standardization, and eco-friendly in nutrient as possible, up to take translation of nanoparticle plant is needed. Thank you for your attention. This work was based on our research from different background. As I mentioned before, I'm from agriculture biotechnology, and my colleague, we have tried to work on this one to find a new cooperation from different background in the agriculture sector. Thank you.
Thank you very much for your interesting research. It's really interesting and worthy, and can proceeds in the economic developments in Palestine. We will move to the next speakers, so we can leave all the questions at the end, just to save time. Okay. Right. The next speaker is Mrs. Ashwaq Najjar. Ms. Ashwaq Najjar has finished her bachelor degree in applied biology at Palestine Polytechnic University, and she moved to do her master degree at Al-Quds University with a scholarship from Agricultural Work Committee. And then her master thesis were done in with a scholarship from the PGSP, Palestinian German Science British, for 6 months under my supervision and Dr. Christina. And now Mrs. Najjar has enrolled as a PhD student in the Institute of Bio and Geoscience IPG2 in Forchungszentrum Juelich in Germany, with the same agency, I mean, through the Palestinian German Science British, with under supervision of Dr. Ahmed Alboraey and Christina Kuchendorf from Juelich, and me from Palestine Polytechnic University. So Ms. Ashwaq will talk about her research as microalgae and biochar agro-fertilization of the Palestinian Rehan, Barley, Cultivar under salinity stress. So Mrs. Ashwaq, it's your turn now.
Thanks, Dr. Sharaf, for introducing me. Can you see my screen?
Yes.
Yes.
That's good. I will start. Okay, good afternoon, everyone. My name is Ashwaq Najjar. I'm a PhD student at IPG2, Forchungszentrum Juelich in Germany. As Dr. Sharaf said, my topic today will be about microalgae and biochar agro-fertilization of Palestinian Rehan Cultivar under salinity stress. As an introduction, I will talk about the Palestinian agriculture status and its challenges. The Palestinian agriculture environment is suffering from high degradation and loss in the agricultural lands. This is due to many problems. First of all, salinity. Salinity is one of the major abiotic stresses factors that decreases crop yield in arid and semi-arid areas. For example, in Jericho, we can see that the agriculture shifted the planting of vegetables to palm trees due to high increasing in salinity levels in the soil in Jericho. Secondly, soil contamination. Soil contamination affects both soil properties and crop yield, especially in quarries and their landfills. Soil infertility exists due to excessive use of chemical fertilizers and pesticides. Also, water scarcity due to the Israeli settlements and low rainwater rate can affect highly the agriculture in Palestine. To solve these problems, our project or our research suggested many solutions to solve part of these problems. First of all, the transfer of nutrients to organic fertilizers. In this regard, we transferred the needed nutrients for plant growth to Chlorella vulgaris algae biomass. The biomass was used as a biofertilizer in poor nutrient soil to support plant growth. This work was done already in IPG2 Forchungszentrum Juelich at 2018 to compare the effect of algae biomass in wet and dry status, which was successfully able to grow up wheat and support its growth in poor nutrient sandy soil. The second solution is biochar. Biochar is a charcoal that is produced from pyrolysis process of biomass in the absence of oxygen. This was firstly discovered in Amazon area, which was called then terra preta soil. This soil was resulted from huge dumping of charcoal with compost, manure, plant materials, etc. So the Amazon infertile soil became a nice green agricultural place. Biochar could change soil properties. So it could change water retention capacity, cation exchange capacity, improve soil structure in general, and increase the activity of microorganisms and soil animal in nutrient degradation. So biochar can be put on the surface of the soil, or it can be mixed with the first layer of soil to improve soil properties and then improve plant growth. Okay, the above mentioned biofertilizers were applied on poor nutrient sandy soil to test the effect of early growth of barley under salinity conditions. Firstly, the effect of different salinity levels were tested on the seed germination stage of different barley cultivars. From the results of this experiment, we chose Rehan barley cultivar as a moderately resistant cultivar. Then, biochar and algae, Chlorella vulgaris biomass were applied on poor nutrient sandy soil under the effect of different salinity stress environment 0, 4, 8, 16 millisiemens per meter square EC. In this experiment, several plant and soil parameters were recorded. Also, our plants were watered and photographed by vessel crane. This is a machine that takes photos from 3 angles to measure the leaf area of the plant by special software for that. Also, another treatment was done to test the effect of the used dried algae biomass to test the comparison between wheat and barley growth. Result and discussion. In this figure, we can see the effect of different salinity levels with biochar and freshwater dried algae biomass treatments. In this figure, we can see that biochar improved both soil and plant growth parameters, but algae treatment didn't. This is maybe because that the first days of our experiments was hot days. We got 35 Celsius degrees in our greenhouse experiment. So it was hard for the microorganisms to decompose the rigid cell wall of Chlorella vulgaris and get the needed nutrients for plant support and growth. Also, we can see that salinity affects plant growth. While salinity increasing, plant growth decreasing. But surprisingly, we can see that biochar at 8 EC could support plant growth even better than its control and even better than the mineral control. After that, to test the bad effect of our results or the negative effect of algae biomass in our experiment, we have repeated the experiment with the same algae treatment. We could find that algae fertilization could support plant growth better than the past, but it's still less than biochar with mineralization addition effect, and also less than the mineralization, the mineral fertilization. Also, another experiment was done to compare wheat and barley growth. Barley showed better and faster growth than wheat under the effect of algae fertilization. The results is not shown in this presentation. Another experiment was done to test the synergic effect of both dried algae biomass and biochar on barley growth and improvement. Our results showed that algae with biochar in one treatment could actually improve plant growth and support its growth, but it's still slower than algae fertilization effect alone, and biochar with mineral addition fertilization alone. This reason still is not clear. We couldn't find any previous studies that explains the mechanism of both effect of algae and biochar fertilization. We still want to find out this result. In conclusion, biochar and dried freshwater microalgae can help and support plant growth even in saline and poor soils. Biochar and dried freshwater microalgae may be a good sustainable solution as a soil conductor or conditioner through the entry of phosphorus, nitrogen, and also carbon. This barley variety is moderately resistant plant to salinity abiotic stress. Salinity-tolerant plant can be a good solution for crop production and yield improvement, which indicate that the used cultivar, Rehan cultivar, is well advised for saline soil in Palestine. In recommendation, more studies and analysis have to be done to understand the degradation process of algae in the soil and also the good timing of addition of algae biomass. This barley variety as a salt-tolerant plant is a sustainable solution for the use of saline agricultural land. There is a necessity to test more during the full lifespan of barley to fully understand the involved mechanisms and relevance, especially in alkaline and saline poor soil areas. The cultivation and production of algae from different sources and also the use of biochar should be further tested and applied in the Palestinian agriculture. Thanks for listening for me, and I'm happy to hear your questions. Dr. Sharaf, we cannot hear you. You are muted.
Okay, thank you very much, Mrs. Ashwaq and Najjar, for your talk. It's really a good and interesting topic, since it is
Dr. Sharaf.
Dr. Sharaf, you are muted.
mute. You didn't hear me anything? سامعيني الآن؟
We can hear you.
We can hear you now.
نعيد مرة ثانية. Okay. Thank you, Mrs. Ashwaq and Najjar, for your nice talk. It's a really interesting topic, since it is dealing merely with the bioeconomy, so we can investment our solar radiation, our climate, which is sunny climate, to produce the microalgae and use them as a biofertilizers to rehabilitation our bad or poor soil in Palestine, mainly in the arid and semi-arid conditions. Okay, we can move for the questions. We can leave them at the end of the sessions in the general discussion. Now we will go to the next speaker, which is Mrs. Hadir Abuwarda. طيب. Hadir Abuwarda has master degree in medical sciences, biomedical sciences, and bachelor degree in biochemistry from Islamic University in Gaza. she was working as a return employee or a return student in the biochemistry department, and she also worked in the research in the genetic labs, and also at the same time in Islamic University in Gaza. So also she will give us a nice lecture about investigating aneuploidy inducing effects of Nemacor, Roger, and Dorspan in human peripheral blood lymphocyte culture. So Hadir, it's your turn now.
Thank you, Dr. Sharaf. The title of my presentation is Investigating Aneuploidy-Inducing Effect of Nemacor, Roger, and Dorspan in Human Peripheral Blood Lymphocyte Cultures. This study was recently published at the Journal of Environmental and Molecular Mutagenesis, and the supervisor of this study is Professor Fadel Sharif. Organophosphate compounds are one of the major components of pesticides. Excessive use of organophosphates is a concerning issue for human health. In the Gaza Strip, organophosphates are the most common insecticide used among farm workers. And during the past 3 years, the most extensively used organophosphate in the Gaza Strip were Nemacor or Finamifos, Roger or dimethoate, and Dorspan or chlorpyrifos, according to the report of the Palestinian Ministry of Agriculture. Although several studies confirmed the genotoxic effect of Finamifos and dimethoate in vitro, no previous studies investigated their effect on the induction of aneuploidy, either in vitro or in vivo. And in the case of chlorpyrifos, there are a limited number of studies that reported the induction of aneuploidy after exposing cultured human lymphocytes to this pesticide. So our objective in this study is to investigate the potential effect of a plain organophosphate pesticide, which are extensively used in the Gaza Strip, on the frequency of aneuploidy in human peripheral blood lymphocyte cultures. And the significance of this study is that finding an association between exposure to organophosphate pesticides and the induction of aneuploidy in human peripheral blood lymphocyte cultures may contribute to the interpretation of the increasing incidence and prevalence of birth defects in the Gaza Strip that result from chromosomal abnormalities such as Down syndrome. Research methodology. This study was performed at the Genetic Diagnosis Lab and the Cell Culture Lab of the Islamic University of Gaza. Firstly, ethical approvals from Helsinki Ethical Committee and the Ethical Research Committee at the Islamic University of Gaza were obtained, and informed consent was taken from the healthy blood donor. And the selected concentration of Nemacor, Roger, and Dorspan are based on previous studies. And the cytotoxicity of this selected concentration on human peripheral lymphocyte culture using the MTT assay was performed to assure that this concentration of commercial formulation which is valuable to us, don't induce excessive cytotoxicity. This table explains concentration of chemicals which we used for in vitro treatments of peripheral blood lymphocyte cultures. We used DMSO as a solvent control and Taxol as a positive control, and Nemacor, 3 concentration of Nemacor and 3 concentration of Roger and 2 concentration of Dorspan. Firstly, the peripheral blood mononuclear cells were separated by a leukobrib lymphocyte separation medium. Then MTT assay was performed either immediately or after 0 time incubation, or after 18 hours of incubation. And the part of investigating aneuploidy-inducing effect of Nemacor, Roger, and Dorspan in human peripheral blood lymphocyte culture using fish technique, we firstly cultured the lymphocyte cells and we incubated it for 24 hours. After this time, we treated it with the different type of pesticide. And after 80 hours, we completed the procedure of fish technique, which harvest and slide preparation, slide hybridization with specific probes. Then analysis of fish signals was carried out by fluorescence microscope, and each culture or each concentration were repeated 2 times, and at least 400 interface cells were scored per slide. Then the frequency of aneuploidy was determined as in this picture. If the nucleus of cells contain 2 copies or 2 signals of autosomal chromosome, these cells considered as a normal diploid cells. And if these cells contain more number of copies of signals of autosomal chromosome, we consider it as a trisomic cell or have chromosome gain. And if these cells contain least number of 2 copies or contain only 1 signal of autosomal chromosome, we consider it as a monosomic cell or have chromosome loss. Data analysis, we used ACPSBS software and the result of MTT assay, and we used the independent sample t-test for the result of MTT assay, and for fish analysis we used the chi-square t-test, and all results were accepted as significant when p-value is less than 0.05. Result of the study. This table shows the cell viability of the human peripheral blood lymphocyte cultures after exposure to different concentration of the 3 types of pesticide, and the result of MTT assays indicated that there is no significant change in the viability of peripheral blood lymphocytes when treated with Taxol or Rogor or Durspan, either at 0 time or 18 hour of incubation. And although a significant decrease in cell viability was observed for Nemacor treated culture, this concentration of Nemacor didn't induce excessive cytotoxicity. Then this table shows the frequency of aneuploidy, chromosome loss, and chromosome gain of chromosome 21st, negative control cultures, and DMSO culture, and Taxol, and the 3 concentration of Nemacor. This table shows some increase in the frequencies of aneuploidy, chromosome loss, and chromosome gain. And moreover, the chi-square test showed a significant difference in the frequency of aneuploidy between treated and the solvent control culture at the concentration of 0.05 milligram per milliliter of Nemacor. From these tables, or this table shows that chromosome loss is present in higher frequencies than chromosome gain for each concentration or each selected or treated concentration. Table 4 also shows the frequency of total aneuploidy, chromosome loss, and chromosome gain for DMSO and Rugor, different concentration of Rugor and Durspan. And this table also shows some increases in total aneuploidy and chromosome loss, chromosome gain in every applied concentration, and also shows the chromosome loss is more frequent than chromosome gain. Discussion, the results showed that 3 types of organophosphate pesticide, regardless of their concentration, led to increase in the frequency of aneuploidy when compared to the solvent control cultures. These results correspond to the results of previous studies that have investigated the ability of organophosphate pesticide in general to induce aneuploidy in human cell lines or cultured human lymphocytes. And in a previous study, exposure of human peripheral blood lymphocyte culture to low level of chlorpyrifos showed a significant induction of chromosome loss. And this finding is similar to what we observed in our study. This observation suggests that aneuploidy may arise from a particular mechanisms of loss rather than a mitotic non-disjunction event. One of the several hypotheses which explain why chromosome loss is more frequent than chromosome gain is the micronucleus formation, which formed due to delays in the separation of centromere. And this corresponds to the findings of several studies that showed an increased frequency of micronucleus after exposure to organophosphate pesticide. Although the observation of micronucleus in our culture was not one of the objectives of this study, our study, we observed an increase in the induction of micronucleus in Nemacore and Rogor treated cultures compared to their respective solvent control cultures. As in this picture, this is micronucleus and this is macronucleus. Another interpretation for why chromosome loss is more frequent than chromosome gain is referred to the limitation of the fish techniques such as signal overlap and hybridization failure. As a conclusion, all the three types of applied organophosphate pesticide induce an increase in the frequencies of aneuploidy as compared to the solvent control cultures, and the induction of chromosome loss was present at higher frequencies than chromosome gain for each examined concentration. And as a recommendation, we need further investigations on a larger number of samples to confirm the association between exposure to organophosphate and induction of aneuploidy and other chromosomal abnormality. Thank you.
Hadir, for your nice talk. It's really interesting one. So wherever there is advantages, also there's disadvantages for these pesticides, or as a residuous to the human and cause this syndromes of aneuploidy. Okay, we can leave the questions at the end of the sessions. Now the next speaker is Yusuf Amr. Yusuf Amr, are you there?
Yes, I am.
Okay. So Yusuf Amr and his colleagues are from Applied Biology departments. They just finished, I mean, fresh. Their talk will be about effects of alkalinity stress on 10 certified Palestinian barley cultivars during seed germinations and the growth stages. So Yusuf, you can start now.
Good evening, everybody. Our project, which as my doctor said, effects of alkalinity on 10 certified Palestinian barley cultivars during seed germination and growth stages. This project will be represented by me and my colleague Khalid Aboushkhedem, which is sitting next to me. Our outline, first we will start with a short introduction about the project, then a problem statement, aims and objectives, the method that we used, results, conclusion, and some recommendations. Stresses affecting plants are external conditions which affect the plant in different ways, especially on the growth and development and the productivity of the plant. These conditions are divided into two main categories: abiotic stress such as high alkalinity and biotic stress such as insects. Alkalinity, which is increasing in the soil pH above 7 due to the presence and the accumulation of alkaline salts such as CaCO3. These salts are introduced into the soil due to many sources, from many sources such as quarries, weathering, and coal-fired boilers. The effects of vegetation, of high alkalinity on vegetation, which is considered one of the most dangerous effects, is considered as one of the most dangerous vegetations due to its changing the soil structure and enzymatic imbalance. As a result, it will inhibit the growth rate of the plant and causing severe cell damage to the plant, especially the roots of the plant. Quarrying is a form of land use method concerned in the land benefit through the stone extraction from rocks with different methods such as rocket drills or explosions. Due to its capital income in Palestine, it is considered, or it's in many countries or in many states in Palestine, it's called the white gold. Quarries nowadays are considered as abiotoxic due to its negative effect on the ecosystem, where it causes air, water, soil, and noise pollution, which as a result will affect ecological balance and damage the biodiversity. Just a minute. And barley, which is one of the most important cereal crops in the world and in Palestine, and has many benefits on a wide scale. And in the medical importance, on the medical perspective, it provides all the nutrients to our body, reduce the cholesterol, and improve the digestion, as well as controlling blood sugar. I will start my video, but due to some complications, there are like sorry. Our problem statement. Due to the high alkalinity stress and many other stresses in the soil, the agriculture has been retreating in the last few decades. Although the large benefit of the barley in Palestine, it is one of the most affected crops from these stresses, especially the high alkalinity. On the other hand, quarries, which are threatening the agriculture and cropland cultivation by introducing the high alkalinity to the soil. So we will explore these different cultivars from barley that can be resistant to high alkalinity of quarries, their landfill, and nearby areas. The main aim of our project is to investigate the effect of alkalinity on Palestinian barley cultivars. The minor objectives of our project is also to investigate the effect of alkalinity on seed germination, early growth stages, and the harvest yield of the plant. We also will explore the cultivars that are most resistant to the soil alkalinity and suggesting a better way to resume the productivity of the quarries and their landfills. Our method in this project, the 10 different cultivars were chosen to be examined under 4 different concentrations of CaCO3, which are 0 millimolar, 50, 85, and 120 millimolar of CaCO3, to study the effect of alkalinity on barley growth and seed germinations. The main steps to this project is soil analysis, media preparation, seed disinfection, incubation process, growth parameter analysis, and anatomical analysis. First of all, we started with analyzing the soil from different sources of soil in quarries in the Hebron Governorate, where the pH as well as the CaCO3 concentration were measured. The seeds of these 10 cultivars were disinfected, then incubated in a tissue culture room for 8 to 10 days, where the seed germination percentage and the G50 of these cultivars were calculated. After that, the seedlings were taken to analyze their growth parameters with the shoot as well as the root length, and the root numbers were measured, and the biomass of this tissue were calculated. The anatomical analysis, a few samples of the seedlings were taken to go under a long process of anatomical analysis, where they were firstly fixed in FAA, then a cascade process. At last, the segments were cut into cross-section and stained with safranine and fast green to be examined under the microscope to study the effect of alkalinity on the anatomy of the seedlings. And now with my colleague Khalid.
Hi, I'm going to start with our results. At the first, we measured the soil, the quarry soil analysis. That are 14 soil analyses. We measured the pH level and CaCO3 concentration, and we found that the highest concentration of alkalinity was 51%, and the lowest one was 33%. While the pH level was, the highest one is 9.7 and the lowest was 8.2. In our results, we study the effect of alkalinity on seed germination percentage. And in overall, we can see, or in general, we can find that alkalinity stress is affected negatively at seed germination. Whereas the alkalinity stress increased, the germination will decrease directly. We can find that Kadawan and Barley, also Axad 176, are the most highest cultivars that their germination was high, in contrast to the rest of 10 barley cultivars, where their germination was about 50%. We study also the effect of alkalinity on G50, which is the number of days that 50% of seeds were germinated. And we found the relation between G50 and alkalinity stress is a positive relationship, whereas increase the alkalinity stress, the G50 also will increase. And we found that Icada-1 has the lowest G50 among the rest of 10 barley cultivars. We study also the effect of alkalinity stress on growth parameters. And I'm going to start with the first growth parameter, which is shoot length. Overall, the length of shoot is, in most cultivars, is constant, and there is no change between cultivated seeds under controlled conditions and the rest of concentrations of CaCO3. While in other cultivars such as Barley, there is no constant, but there is an increased and sharp increase in its shoot length. About the length of root, also there is no very change in the most of cultivars, and there is some exceptions such as Rehan, where there was steadily decrease in its root length, while Barley, in contrast to Barley, where its length of root was steadily increased. About the average number of fruits, most of cultivars, or to say that most of them or all of them have roughly the same number of fruits, and this indicate that the barley could be resistant to alkalinity stress without increase its number of fruits. And you can see that the most of them have about 5 roots. This graph implies the alkalinity effect on dry weight, which is a biomass, as we know. We can see that there is a change or unsimilar results, and this related or due to the different ability of the cultivars to respond to the alkalinity. So as the alkalinity, when the alkalinity change or affect the cultivars or plants, most of them have a different ability to respond to them. So we can see this difference in most results. We can see that Cada1 have roughly the highest dry weight, while improved Barley, in contrast to control cultivation, there is a really high difference in its dry weight. The dry weight also in root, the same thing, which represent the ability of plant to respond to stresses, such as abiotic stresses. Total water content is complementary to dry weight, where in Ecodo 1 should be back to Ecodo 1. Ecodo 1, its total water content is about 30%, and if we go back to its dry weight, it's about 70%. So that proves our theory about the complementary theory between them, between the total water content and dry weight. Also, the improved Baladi roughly have 50%, and its dry weight was also roughly 50%. As well as the total water content of the root, the same thing that as I mentioned before about the shoot system. Due to our preliminary results and during the anatomical process analysis, we found that Ecodo 1 was the most resistant cultivar among the Western barley cultivars. The pictures in the left here is for the Ecodo 1 roots under control conditions, and we can see that there is no effect at its root when it's cultivated under 120%, 20 millimolar of CO3. And this again improves our results that Ecodo 1 is the most resistant cultivar. In conclusion, alkalinity stress is one of the most abiotic stresses that threatens the seed germination and growth stages of the certified 10 Palestinian barley cultivars. Also, Ecodo 1 is the most resistant one during the seed germination due to high seed germination at 120 millimolar of CO3, low G50 time, and high dry weight to water content ratio. And improved Baladi cultivar is the most sensitive cultivar between the rest cultivars. On the other hand, most cultivars of barley are resistant to high alkalinity even at 120 millimolar of CO3. Our recommendations: we recommend one, further research to explore the mechanism action of alkalinity CO3 on vegetation. Two, to integrate the tolerance species in a roadmap of rehabilitation of quarries, as well as to reassess the legislation for quarrying in Palestine in the frame of mitigation the pollution from quarries industry. Thank you for listening. And can I have any question? Yes.
Thank you, Mr. Khalid and Mr. Yusuf, for your nice presentation. It's very interesting research, since it gives model plants for cultivation in poor soil of quarries to resist the high alkalinity, which is almost around 9. The pH is almost around 9. So we now move to the next speakers. So you stop sharing, Khalid.
Thank you.
But you have to stay with us at the end of this sessions, Khalid and Yusuf. Okay,
yes,
don't leave.
Yes, sir.
Okay. So the next speaker is Ghuslan Awad and her colleagues. And they are also from Applied Biology departments at Polytechnic University. Her presentation is in vitro evaluation of biochemical parameters for selected and certified barley cultivars grown under salinity and alkalinity stress. So, Ghuslan, you can start now. دكتورة غدير.
عفوا. دكتور.
غزلان يعني
ضفته.
she able to غزلان.
He's panelist. هيو.
معود موجود.
محطوط panelist دكتور غزلان.
حرف الجي.
محطوط حولته دكتور شرف بس مش عم برد.
غزلان
ابعت له على الشات مسج.
هي موجودة هون. اوكي.
هي وحدة ولا واحد؟
واحدة.
طيب اوكي. اسفة بس كان عندي طالب دكتور.
No problem.
انا حولته لبانيليست. بعرفش اظني بتقدر انت كهوست تبعت له اشي كليك هيها اوكي غزلان شكله النت عم بفصل عندها كمان في عبد الله ابو زينة اذا موجود يرفع ايده عشان اشوفه
موجود
حولته converted to panelist
خلاص اوكي محلول الموضوع فيش مشكله.
اوكي شكلها غزلان عم بفصل عندها النت.
وين هي غزلان؟ اجت هون هي؟ من الصبح بنحكي.
وينها؟ هلا كانت شكله فصل عندها. لا هي موجودة.
دكتور خذ اللي بعدها اذا في اشكاليه.
دكتور عبد الله موجود؟ ايوه ناخذ اللي بعديه.
طيب خلينا ناخذ اللي بعده. طيب the next speaker for this session is Abdullah Abu Zena and Dua Hayes. They are also from Applied Biology Department from Palestine Polytechnic University. They will give us a lecture about their research projects entitled Synergic Effects of Salinity and Alkalinity in Certified 10 Palestinian Barley cultivars during seed germination and growth stage. So you can start now, Abdullah. I will yeah. Okay. ان شاء الله انك ما تسوي لنا اشكاليه هذا الحين.
سامع برد عليك ولا؟
لا شغال. هلا ظهر من عندي انا البرزنتيشن تبعته. اوكي
ظهر.
Okay, so Dr. Abdullah is
Hello everyone, my name is Dua Ibhis and my colleague Abdullah Abuzene. Today we will talk about our project about the effects of salinity and alkalinity stresses on Palestinian barley cultivars during seed germination and growth stages. In introduction, we will talk about plant stresses, salinity and alkalinity stresses, sources of salinity and alkalinity, the effects on vegetation and barley. Then we will talk about aims and objectives, methodology, results. After that, we'll talk about discussion and conclusion.
Good evening, everyone. Plant stresses. Plant face a lot of stresses. Could be biotic stresses or abiotic stresses. Biotic stresses like viruses, bacteria, insects. Abiotic stresses like cold, salinity, alkalinity, dryness. So, what we mean about salinity stresses? Salinity stresses is the accumulation of salt in soil. Could be from chemical fertilizer or have evaporation. And alkalinity stresses is the increase of the pH in soil. Could be from quarries or alkaline compounds. The effect of salinity and alkalinity on plants. Its effect in different ways. Could change the osmotic pressure. Could cause ion toxicity and increase the pH level. In this project, we choose barley. Why we choose barley and what's barley? Barley is angiosperms and monocot plant. Why we choose barley? Because it can grow in moderate pH, have a short life, and it's considered like a resistant plant for salinity and alkalinity. The aim and objective in this project to see the most resistant cultivar to salinity and alkalinity stress, and improve the production of area near quarries. The plan we followed in this project divided into two sections. Section number 1 was soil analysis. We collect 16 samples from different 3 areas in Hebron. We measured the pH and the electrical conductivity and the percentage of CaCO3. Section number 2 was seed germination. We were provided by 10 cultivars from North Center. We prepared the media for different concentration. Step number 2 was seed disinfection to make sure there is no contamination. Step number 3 was seed planting and cultivated in culture room. And the final step was morphological and biomass analysis. The parameter we chose in this project was 9 parameters. We choose seed germination. For shoot system, we choose the total water content, the length of the shoot, and the biomass of the shoot. For root system, number of roots, total water content, and biomass and length. This is the standard deviation for soil analysis from the 3 different areas. We collected the sample from Bani Naim and Ashwaq and Sair. And this is the whole schedule from the 16 sample. The result of 16 sample. And I'll leave you with my colleague to
add. لو سمحتي
بتغيرش العرض عندكم
لو سمحتي سمعتيني؟
ايه تفضلي.
السلايد مش عم بيغير له من اول البرزنتيشن على نفس السلايد الاولى، وبعدين اعملوا سلايد شو عشان تبين كلها، كل اللي حكاه ما غير ولا شريحه، معلق. فهمتي علي؟
لو سمحت عيد شوي.
السلايد شو اللي عندكم محطوط على البرزنتيشن على اول سلايد، لما كان يكبس مش عم بيغير، وحوليه لسلايد شو مش زي ملف هيك. يعني كل اللي حكاه احنا عنه مش ظاهر انه غير.
اول شيء يعملوا enable editing.
ايوه اعملوا enable editing.
عيدي بس سريع. خلص احكي انت من جديد. هيك بغير هيك؟
هيك تمام.
كيف؟
وصلنا ريزلت.
طب ما بوصل الصوت.
مش الصوت
مش الصوت يا دكتور. السلايد
طب خلاص كملوا.
كملوا الريزلت احنا باش نهاية.
طب كملوا يلا.
The 10 cultivars of barley was germinated at 4 different concentrations and 3 specimens for each concentration. The sum is 120 plates. In this graph, show that the total percentage of seed germination. As the concentration increase, the seed germination percentage decrease. The average time of G50 is the time that needed for 50% of seed germination. The average number of fruits. In this graph, there is 85% of results are between 5 and 6. The average length of shoot system. Generally, as the salinity and alkalinity stress concentration increase, the length of fruits increase. The average length of shoot system. As the concentration increase, the average length of shoot decrease. The percentage of biomass of root system. Dry weight, it's referred to the weight of tissue dried in oven at 60 Celsius. The percentage of biomass of the shoot system. As the biomass of shoot increase, it refers to high rate of photosynthesis rate. The total percentage of total water contents of root system. It's increase the amount of water in the root in order to resist the osmosis resulting from the accumulation of ions. The total percentage of total water content of the shoot system. The water content is the amount of water contained in the material. In discussion and conclusion, the response to salinity can differ among barley cultivars. For example, Rehan considered a resistant plant due to high percentage of seed germination, appropriate average G50 time, shoot length, and biomass stability in high concentration and high total water content of root system. On the other hand, Axed 1417 seems to be sensitive cultivars due to its low percentage of seed germination, no G50 decreasing of its shoot length and biomass. While Icarda1 is a moderate with moderate percentage of seed germination, a reasonable average of G50, moderate percentage of a dry biomass in the shoot system, and high total water content in root system. In conclusion, the result shows that there is a certain cultivars can be resistant to stability and alkalinity stresses that can be cultivated in nearby area of aquarius. Our recommendation, the highest biomass is good to use as animal fodder. The maximum percentage of seed germination will be suited for economic benefits such as human commerce. The lowest average time of G50 could be used for Palestinian farmers. Thank you for listening.
Okay, thank you, Ms. Dua and Mr. Abdullah for your nice presentation. So we can now move to the next one. Ghazlan Abu Awad. Do you hear us? Ghazlan موجودة؟
موجودة.
Okay, share your presentation first. طيب لين الماضي موجودة؟ لين الماضي
مش موجودة. موجودة؟
لين الماضي موجودة؟
نو حتى لا بالاتنديز ولا هذا.
طيب غزلان مضيوفة على البانل ليست؟
يس شي از
موجودة
زمان.
طيب غزلان share your presentation.
مش عم بترد اصلا، حتى هذيك مرة نادينا
اسمح جوال.
لين
دكتور ننتقل للسكشن اللي بعديه. دكتور خالد صبارنا. دكتور نهال موجودة؟
نهال لا مش شايفها.
أمريكا.
الوقت ما صارش الوقت بالنسبة للدكتورة نهال لسه.
لا لسه مطول، ما في بريك عنا بعد هاي السيشن.
دكتور يعني تعبنا لازم ناخذ بريك، بس بالأول نشوف يعني إذا نجحنا مع غزلان أو لين. اوكي اذروايز بناخذ جنرال ديسكشن وبعدين ناخذ لنا بريك يعني نرتاح لنا شوي. ف غزلان احكي مع غزلان تليفون. معك رقم غزلان؟
ايوه
زبطت. اوكي جود.
مساء الخير.
مساء النور غزلان. Our presenter now is Ghazlan Abu Awad. She is applied biologist from Palestine Polytechnic University. Her presentation is in vitro evaluation of biochemical parameters for selected and certified barley cultivars grown under salinity and alkalinity stresses. You have 10 minutes Ghazlan.
تكبر الشاشة عندها.
مش مشكلة. Go ahead.
My presentation is about the in vitro evaluation of biochemical parameters for
شرف مش واضح، السلايد كتير صغيرة يعني. واحد
غزلان
ما بعرف ايش المشكلة في طريقة العرض.
خلص مش مشكلة بنمشي، مش راح يزبط تكبر يا غزلان. انت اوكي good now it's okay. يلا.
In vitro evaluation of biochemical parameters for selected barley cultivars growing under salinity and alkalinity stress. Outline, I will start with the introduction, talking about queries, salinity, alkalinity, barley, and biochemical parameters, aims and objectives, methodology, result and discussion, recommendations. Quarries, stone and marble industry in Palestine, locally named as white gold, is considered as the main extractive industry in Palestine.
concentrated mainly in Nablus, Jerusalem, and Hebron cities. And Palestine is ranked 12th worldwide due to its contribution by 4% of the world's production of stone and marble. In quarrying area, the soil attributes to the high concentrations of hydroxyl, carbonate, and bicarbonate ions, and these pollutants cause an alkalinity nature with a pH range between 11.2 to 11.7, and it's also the primary minor mineral rocks cause of all the dissolvable salts present in the soil, and so it makes the soil saline, and it can raise the electrical conductivity of the soil to more than 4 AC in many cases. Salinity, it is one of the major abiotic environmental stresses and the most widespread land degradation problem. And the soil affected, saline affected soils occupies on global basis large areas, and these soils constitute nearly 7% of the total land area, or nearly 33% of the potential agricultural land of the world. Alkalinity, soil alkalinity, alkaline soils occupy a higher percentage than saline soils around the world, and it is characterized by the accumulation of mixture of sodium bicarbonate and sodium carbonate salts in the soil. Also, the injury that results from alkaline conditions are more pronounced than those caused by salinity, and it includes cracking the soil structure due to strong effect of sodium ion, low calcium absorption, carbonate ion toxicity, and caustic effects of high alkaline. Barley is the first most significant cereal plants in the world after wheat, rice, and maize, and it is the most cultivated one. In Palestine, barley cultivation occupies 28% of the cultivated area with field crops, and it is tolerate drought more than wheat. One of the ways for the reclamation of alkaline and saline soil is by the cultivation of tolerance plants, and barley is considered moderately tolerant plant since it's among the cereal plants. It is the most tolerant to salinity stress, and it is can tolerate alkaline and it can grow in alkaline conditions when a pH values range between 6 to 8.5, and it also can tolerate highly alkaline soils more than any of the other small grains. So biochemical parameters will measure to determine the effect of salinity and alkalinity in barley. Biochemical parameters, chlorophyll fluorescence. The chlorophyll fluorescence measurement is relatively a new technology that has recently been used to study the effect of various stresses on plant, on photosynthetic efficiency of the plant. These chlorophyll fluorescence parameters can be used to monitor the growth, the physiological response, and photosystem 2 changes in plants under stress conditions, and the total chlorophyll fluorescence amount is only 1 to 2% of the total light absorbed. Fluorescence provides a good, rapid, and invasive measurement of changes in a particular sample through time, and it can be also useful in examining the acclimation of the plants under different environmental stresses. Since the damage to photosystem 2 considers the first manifestation of any stress on a plant, chlorophyll fluorescence parameters can provide information about the state of photosystem 2, telling to which extent it is used the energy that's absorbed by chlorophyll molecules, and the flow of the electrons through photosystem 2 is an indicator of the overall rate of photosynthesis, and hence it allows us to estimate the overall photosynthetic performance. The chlorophyll fluorescence parameter we study in this project is the quantum yield, which used to indicate the efficiency of photosystem 2 photochemistry by measuring the proportion of light that is used by the chlorophyll molecules associated with photosystem 2 that is used for photosynthesis, and it can also be used to calculate the linear electron transport rate and therefore the overall photosynthetic performance. The chlorophyll content, as we know, the chlorophyll is the most important class of photosynthetic pigments that aim to convert the light energy to chemical energy by absorbing the energy from light and use it to convert carbon dioxide to carbohydrates, and it is play a unique role in the productivity, physiology, and economy of the green plants. And its amount in the leaf varies according to the nutrient availability and by environmental stresses such as drought, salinity, cold, and heat, and etc. So it's important to measure the amount of the chlorophyll to see how these factors influence the chlorophyll content in the plant leaf. The relative water content is basically related to the state of water balance in the plant, since it expresses the amount of water that is needed by the plant to reach full saturation, and this positive trigger is necessary for plants to maintain photosynthesis under stress conditions and for cell expansion in growing tissues. So the relative water content provides a measure for the water deficit in the leaf, and it can indicate the degree of stress that is expressed under exposure to various stresses, including those of salinity and alkalinity. The normal values for relative water content range between 98% for the fully turgid leaves and 48% for the severely desiccated and dying leaves. Leaf extract pH, the stable tissue pH, as a result of intracellular ion balance, is necessary for the plants to maintain their normal metabolism. And for in a living plant, as long as this plant can adapt to its environment, the leaf extract pH are assumed to be stable regardless of the changes in the pH values in the surrounding environment. Now the problem statement. There are two kinds of stresses that are exerted on a plant cultivation, biotic and abiotic stresses. And recently, quarries are considered as a cause of abiotic stress due to their effect on air pollution and increased salinity and alkalinity of soil. So exploring plants that are capable to compromise with these stresses has received great interest. In this research project, we aim to evaluate the rates of barley during the early growth stage for their resilience to high salinity, alkalinity, and both of them. Biochemical parameters such as the leaf extract pH, relative water content, chlorophyll content, quantum yield will be measured. The main aim is to investigate the efficiency of Palestinian barley growth and development under salinity and alkalinity stress. And the main objectives is to investigate the mechanism.
غزلان try to finish يعني انت لغايه الان في aims and objectives. يلا hurry up.
To investigate the mechanisms of stress tolerance throughout measuring the biochemical parameters of selected barley cultivars and to determine which cultivars are suitable for cultivation in areas suffering from abiotic stress. In methods, we choose 3 barley cultivars, the most 3 barley cultivars according to the results obtained by other studies. So we choose the most sensitive, resistant, and moderate affected cultivars from each treatment, and we use it in our study work. The selected seeds were germinated in wet tissue paper and covered with aluminum foil and incubated at 25 degrees Celsius for a week in the oven. After that, we prepare the alkali solution by adding appropriate amount of calcium carbonate to measured amount of distilled water determined according to the water holding capacity of the soil, and we also prepare the fertilizer by adding 1 ml of solution to 1 liter of distilled water. Then we planted the 2 of the germinated barley seeds in 3-liter plastic bottles and watered them firstly with the alkali solution or with the distilled water for the control plants and with appropriate amount of the fertilizer, and then we water the plants every 2 days with tap water, with gradually increasing the amount of fertilizer during the experimental period of 1 month. The measurement of relative water content was obtained by determining the fresh weight, dry weight, and turgid weight of the leaves, and then calculating the relative water content according to this equation. Measurement of leaf extract pH by homogenizing appropriate amount of the leaves with measured amount of distilled water and centrifuging it, and then measuring.
Try to go to the results. You are running out of time.
Chlorophyll content measurement by SPAD meter. It's a portable meter without and not non-destructive like the extraction procedures. Finally, measuring the quantum yield with the fluorobin device. Rather than discussion, we find that the chlorophyll content of Akasad and Acarda cultivars are not significantly different from the control, and this may indicate the ability of these cultivars to maintain ionic balance and prevent degradation of photosynthetic pigments, while improved Baladi cultivar, its chlorophyll content decreased with the increasing in the intensity of the alkalinity stress, and this has been attributed to magnesium precipitation that will inhibit chlorophyll synthesis, and also due to the activation of the chlorophyll degrading enzyme chlorophyllase, or due to the deficiency of specific minerals that are involved in the synthesis of chlorophyll, or due to the effect of the alkaline stress and decreasing the expression of certain proteins and genes that are involved in chlorophyll synthesis. The quantum yield of the 3 cultivars decreased by varying decrease as the intensity of the chlorophyll, as the intensity of the alkaline stress increased, and this may be related to the variation in the susceptibility to photoinhibition between the three cultivars, since the alkaline stress cause reduction in the activity of photosystem 2 by the combined effect of the high pH and the high sodium influx. But Akasad cultivar increase its quantum yield at 120 millimolar, and this may indicate that it is developed some protective mechanisms such as increasing the heat dissipation to prevent damage by excess light under alkalinity stress, so or by other mechanisms. The relative water content decreased for improved Barley cultivar, and this may be related to the osmotic stress that results from water loss or the difficulty in absorbing it, which will lead to less turgor and reduce the water availability that is required for physiological processes and causes early senescence. However, it is still significantly high, and since relative water content is the most direct reflection of stress tolerance of degree of injury to seedlings under alkalinity stress, we can conclude that the ability of improved Barley cultivar to maintain it is the osmotic balance under alkalinity stress. The other two cultivars, Icarda and Akasad, success to maintain high relative water content, and this will help us, will help them to maintain the physiological balance by increasing the transpiration rate, and will enable them to tolerate to alkaline stress by making osmolytes with low energy consumption. Our results show that the alkalinity stress different intensities. Okay. That the different intensities of the alkalinity stress does not affect the pH of the three cultivars, and this can be attributed to the ability of Barley cultivars to maintain ionic balance and stable pH in cells under alkalinity stress. So in conclusion, we find that the improved Barley cultivar is the most sensitive cultivar, while Icarda is moderately tolerant according to these parameters. And Akasad is the most resistant due to the high relative water content, chlorophyll content, and the quantum yield. And basing on this, we highly recommending it for cultivation in quarries their landfills and nearby areas. And in recommendation, more research is needed to determine the impact of alkalinity on various Palestinian national crops. Field experiments in non-alkaline soil areas are needed to ensure the tolerance of these cultivars to poor alkaline soils, and there is a necessity to assess other chlorophyll fluorescence parameters along with the quantum yield, and to have more helpful information about the activity of photosystem 2 under alkaline stress. Thank you.
يعطيك العافية غزلان. يعني بدي احكيها بالعربي، يعني كان لازم عاملة بروفا أكثر من مرة حتى يكون الوقت مناسب يعني للبرزنتيشن. أنا بعرف الشغل كثير كويس وفي نتائج كثير كويسة، يعني حبذا لو أنت عملتي يعني بروفا بحيث إنه يكون الوقت على القد. طيب إحنا الآن اعملي غزلان اعمل كلوز او ستوب شيرنج. طيب احنا حاليا اخر متحدث اللي هو لين الماضي برضه مش موجوده معنا اليوم، فعلى ما يبدو في عندها اشكاليه معينه. باسمكم جميعا بنرحب بالدكتوره نهال ابو الليل لانها دخلت قبل شويه على الميتنج.
شكرا لكم.
صباح الخير بالنسبه لكم انتم. احنا
صباح الخير.
طيب الان احنا في عندنا جلسه نقاش general discussion for this sessions, And it may be the largest sessions, this one, because we have many talks. And okay, Dr. Christina وأحمد وأشواق وهدير ويوسف وغزلان وعبد الله أبو زيني يعني يكونوا موجودين معنا عشان for discussion. It's really interesting topics. This what we heard today about plant biology, since it is correlated directly to the bioeconomy. So Dr. Christina has good experience in this investment, how to use natural resources and convert the waste products to a resource again. An expression is really interested to, which is a green resource. So microalgae to be used as a green resources for human, and we can visit the Middle East environments or climates, which is a shiny little, so we can grow up microalgae to produce large biomass, and then which is can be converted into biofertilizers to be used as substituted for organic fertilizers, manure, which has had many aspects, I mean wrong aspects, such as spill bad and insects, rodents, etc. So it is a good idea to be used as biofertilizers, and other models of plants have been used in the other research of these sessions. So to be to test which is barley cultivars are resistant to salinity, which is resistant to alkalinity, and in both of them, salinity and alkalinity, and to test the last one with Ghazlan to test the biochemical parameters such as total water contents, biomass, chlorophyll contents, ascorbic acids, etc., under the effect of such stresses. So also Mrs. Hadir, which is also stressed on the stresses, how the stresses can affect the aneuploidy of the human, I mean, in the peripheral blood cytogenesis or lymphocytic clusters. So it's obvious that the stresses can produce bad effects on life and the quality of the human being life also. So it is good to think altogether how to change or rehabilitate, to rehabilitate the soil, poor soil, from different aspects to make it productive. Okay, so if there's any questions from the attendants and all of you, or if somebody have an idea or to participate.
And I want to ask Yusuf.
Yes.
Yes.
Please, during your presentation, you provided us with an anatomical analysis. Okay.
Yes.
I didn't get exactly what is the difference in the two tissues that proved for you this evidence as a result in your study. You said that the alkalinity stress affected the anatomy. Okay.
Can I share the screen?
Yeah, please. You are allowed to share screen, please.
Yeah, just a little.
Because from the image, I didn't see the difference between the two sections.
Just a second, please. Sorry. We use the Icarda in this anatomical analysis. The use cultivar is Icarda, which is the
slide. Yusuf, كبر الشريحة, please expand the slide, make it.
Sorry.
Okay, what is the difference between the two sections?
In these sections, there are no differences because the Icarda واحد, as we said, or Icarda 1, as we said, is the resistant cultivar. So if we can compare, or if we compare between the anatomical sections, there are no big difference. If you see that the tissue lining is not أو فيش اختلاف، إذا ممكن أحكي بالعربي، إنه فيش اختلاف في عدد أو صفوف الخلايا اللي موجودة. يعني مثلا إنه في بعض الحالات سبب ربتشر عندنا، فبالتالي خربت الخلايا المحيطة بالأوعية الناضجة. نعم.
Yes.
في الأوتر layer.
Oh.
Yeah, I can help a little bit in this point. Since there is no significant difference in the anatomy at low concentration or control 0 millimolar in CaCO3, and that one of 120 millimolar, the plant. So if we increase the salinity, the plant can resist this change. So there's no difference in the anatomy.
No difference.
And however, other cultivars, there is a rupture in the cortical cells around the endoderms of the roots, and this can be interpreted to break down the water column due to through the apoplastic and symplastic pathway.
Oh, okay.
So yes, this is might be an adaptation mechanisms to the plant to mitigate the uptake of salt from the soil.
Mm-hmm. Okay. Okay, thank you.
Any questions? Dr. Christina, دكتور حنان.
دكتورة حنان لا موجودة.
Yeah, I'm here, but I'm just يعني enjoying what I'm hearing. I don't have any question.
Okay, so
I maybe don't have a question, but a note that is about something which we would like to look into, but it's not really on our agenda, and that is how, for example, materials that we add to the soil, if it's, for example, biochar or algae or manure or something, how that can change actually the effect of the salt content of the soil if we have a more saline soil. So there is already research for some changes, for some additions to soil, but not much for these newer soil amelioration substrates. And that would be interesting to follow that more up in the future. So no, there is not a project yet, just to leave the thought. Yeah. Also for longer time periods, so not just in the laboratory, but actually on real soil and real substrates in a slightly larger.
Christina, since you mentioned this, are there any studies in your research work at the genetic level, at the gene level, regarding these microalgae and the barley that are salt tolerant?
Mm-hmm. Can you repeat that? I just something was babbled before genetic level.
Yeah, I don't know. I'm wondering if you have any studies at the gene level regarding these microalgae or the barley cultivars that are resistant to soil and how they are interacting with the soil microbes, for example?
I leave the barley to our Palestinian partners because that was where the choice came from. But for the algae and the microbes, we are actually kind of in the middle of doing this because we are trying to analyze when we have a culture of algae and bacteria, because it's never sterile, they all live together. We try to see at which point in time do which functional groups of bacteria turn up in these cultures. And for example, one thing we have found is there's a high amount of alpha proteobacteria groups where rhizobium is, for example, a member of, and this is even a plant growth promoting strain of bacteria. So we do not know if this goes further and might be an additional boost in this, because there's still the transfer. This is when we are still in the algae culture. It has not been transferred to soil yet at this point, but this is also something which could be followed, because in the one place, it is already a working ecology between photosynthetic cells and microbial cells. And then this is transferred somewhere else. And actually also many of the algae which we have, they do not only live in water, they can also live in moist soil. There's much which is not yet really known. And then when we put them in the soil, of course, they are degraded. And we do, for example, not yet know what is the main degrading organism. And this would be important to see how quickly are the nutrients released. Because so far we are observing that the nutrients are transferred to the plant. We also know that it's the nutrients from the algae that are transferred to the plants. We have made radioactive labeled experiments to do this. So we have fed the algae with radioactive phosphorus and then later checked if the plants were radioactive, which they are. So that is that worked quite nicely. But for the exact process, like what has to happen in the soil that nutrients are released in a certain form to the plants, and if it is an advantage, then they are not lost into the leaching water from the soil, but they go kind of directly from the biomass in the rhizosphere to the plant. So I can imagine lots more experiments to do this.
Yeah, very interesting. Very interesting, really.
I just wanted to follow on what Christina and Hanan mentioned, and just like for maybe just brainstorming here and future collaborations. My lab is also interested in how additives to soil, such as biochar specifically, can interact with bacteria within soil systems. My lab is more interested in not plant biology, but we look at that from an environmental aspects of controlling the attenuation basically of bacterial pathogens in the subsurface. And then we are interested in using a variety of different biochars at different sand to biochar ratios, and understand fundamentally how the bacteria or different strains of bacteria that go all the way from non-pathogenic to pathogenic can transfer in the sand or soil columns. We vary everything from acidity to salinity, to sand to biochar ratios, to biochar chemical modifications. We make all the way positive to all the way negative, to like dope them with different components or chemicals. And we do our studies not at the genetic levels that you guys are talking about, but we are interested more in a transport mechanism. How do they affect the transport within columns of mixed phenomena? And we do also a little bit of atomic force microscopy to quantify the forces that govern the interactions between a biochar and a microbe. And we also do some kind of modeling with colloidal theories, like how if we consider the bacteria as a particle and the sand as a particle interact together. So just like touching points, we might have some connections in these things.
That is indeed a really good touching point, because we are going to do some larger scale experiments, which of course will not be so finely structured, but exactly these effects will be turning up there. So yes, let's keep that going.
Yeah.
Okay, thank you, Dr. Nihal. And I think there is a common interest between all of us. So we aim at the end of this conference to have a new cooperation. So we can think about by trilateral projects altogether. It is working. Dr. Christina, do you think it's worth it to raise up the microalgae from economic point of view to be used as a substitute for manure or organic fertilizers? I mean, the cost of the production of microalgae, since microalgae not there is no mass production of microalgae since they are microorganisms.
Yep. I think this will be again a kind of a combined or complex solution. For example, if you have a farm and you use the algae to clear up water, you will end up with additional biomass. And this you can use as a high quality fertilizer. If you would solely aim to just produce algae biomass for fertilization, that will be a rather expensive fertilizer. So there has to be something additionally has to be happening to make this really feasible. So you have to, like I showed before in this picture with the many resources, the algae can be connected into several parts of this, but to get this really to a balance, you kind of have to use everything which they have to offer. So you maybe, if you have a more expensive trade, you also have to be able to take a small portion, extract something more expensive for value for this, as long as the, because there is a kind of critical size of this application, you have to have a large enough facility to have a significant amount to use on the field for a significant positive effect. And as long as you do not have this, it is too expensive. So, and there is not enough experience yet for this kind of closed cycle with an algae fertilizer. But there is already lots of experience, for example, with biochar addition and how expensive is that to get, for example, from a biogas plant and then reform it into the char station. So this kind of experiences needs to be combined with this. So it's still a rather complex thing, but yeah, promising, especially from if you take into account that this is a way to try to form a soil, a substrate for a longer time. So it should not be viewed as a one-time fertilization, but as an addition into the soil where you profit for a longer time period, hopefully, because also the carbon is taking part in this, which you add into the soil. It complicates the calculation, but it heightens the interest.
Okay, thank you. If Mrs. Hadir, is it with us?
أنا هيني
Hadir.
Hadir, okay.
Yes.
It's not, it's in Arabic.
Okay.
Hadir, I have a questions for Hadir, but yes, there is. Hadir Abu Warda.
Yes.
Okay. I would like to ask you, since your research was on Dorspant, Roger, and
Nemacor.
These, what's their effects in human health? And I know that the farmer are using them for example, to treat grapes, and the grapes is the major crop production in Palestine. So do you propose to stop using these pesticides, or what we can conclude from your talk?
We recommend to minimize the excessive use of this pesticide, especially in the Gaza Strip. These 3 types of organophosphate extensively used in the Gaza Strip without any control from the government.
Yeah, but internationally are forbidden to use them, or just in Gaza or in Palestine in general? Since as far I know, it is permits to be used.
Yes, there is a limitation about using this pesticide.
It is a serious problem?
There is several studies found an association between excessive use of this pesticide and several human health problems such as cancer and Alzheimer's disease, Parkinson's, and birth defect. So our objectives is to investigate their effect on the induction of aneuploidy in human peripheral blood lymphocytes, and to find what's effect to induce aneuploidy, which is a type of chromosomal abnormalities in human peripheral blood lymphocyte cultures.
Okay, you mean excessive use of these
That's
pesticides,
excessive
not
use.
a normal one. And you have to pay attention for the safety period. So how many days after we spray them on the products, we have to wait until to be safe to harvest our products? Yes.
I don't know about this.
فترة الأمان بنسميها بالعربي فترة الأمان. يعني احنا بنستخدم هاي المبيدات بس في فترة أمان أسبوع 10 أيام حسب المسحوق اللي بنستخدمه احنا أو المبيد.
صح.
فترة الأمان يعني بتكون أقل الإفكتس تبعها، إذا استخدمناها مباشرة الإفكتس بزيد يعني. Okay, if there is any questions or anybody would like to comment. So otherwise we have from okay.
في حدا بعد سؤال على based on the type.
أحمد.
إيش؟
Based on the type of.
أقصد فترة الأمان يعني تكون مقصودة على نوع المبيد. حسب المادة بتاعته بس حبيت أضيف شغلة بس.
Okay, thank you, Mr. Ahmed, Dr. Ahmed. If there is any comments, so we can leave now. Okay, first, before leaving, I would like to express my deep gratitude for everybody in these sessions because it's really good one, rich with informations, knowledge transfer. So thank you, Dr. Christina, Hadir, Ahmed, Ghuzlan, Yusuf, Khalid, Abdullah, Dua, everybody who are participating in this sessions, and also my colleague Ghadir for managing these sessions also, and Dr. Hanan Malkawi, everybody in here. So we can, we will have a break, just 10 minutes. Okay, so we can come back to save time. I mean, within 10 minutes, we can come back to start the new sessions with Dr. Khalid. Dr. Khalid Sabarna, prepare yourself for after 10 minutes. Okay? Okay, thank you, everybody. So me and Dr. Ghadir, we can stay